Hey folks, it's me, Mr. Lang, your friendly neighborhood AP biology teacher, and today I want to prime the pump for later on in the week when we see each other for the photosynthesis experiment data collection, and it really is the simplest of designs. So I want to start by talking about photosynthesis. You know some things about photosynthesis because you've read about it and you've watched videos where I was standing or sitting up in the corner talking about photosynthesis many times now. So you've got a good background in photosynthesis. So I want to just go through a couple of ideas. So first, I want you to ask yourself, what do plants need for photosynthesis? Think about it. There's three things. If you've only got two, keep thinking. Okay, so plants need carbon dioxide. We know they need carbon dioxide. Where's that carbon dioxide going to be used? Think about it. It's going to be used in the Calvin cycle because we need a source of carbon to make glucose. So that's where the carbon dioxide is going to be used. We also need water. Where's that water going to be used? It's going to be used in photosystem 2, which comes first, remember, and it's going to be split in the chloroplasts. We also need sunlight or a source of light of some sort, and really what we need is a source of photons so that the photosystems can energize those electrons and we can harvest that energy in the form of ATP or NADPH. So those are the three things that we need. Now, I need you to ask yourself, which of these three things is the easiest to manipulate? Because when you're doing an experiment, we could manipulate the amount of carbon dioxide, but that's going to be kind of hard. It's all over in the air, and figuring out how much carbon dioxide we've got is sort of tough because carbon dioxide sensors are kind of notoriously faulty. So carbon dioxide is not going to be the thing that we're going to manipulate. Plants are mostly made of water, and the amount of water that's in a plant depends on the uptake, and if you've got a water source in the experiment, you don't know how much water is being taken into the plant, so that's hard to mess with as well. So sunlight is going to be the thing that we're going to manipulate. So we'll have a setup in shade and have a setup in sun. Now, ask yourself, what do plants produce through the process of photosynthesis? Think about it. They make oxygen, that's sort of a byproduct of the process, and they make glucose, which is what we need out of the process. Although I guess, yeah, we need oxygen too. So which of those things is easier to measure? Well, glucose level in plants could absolutely be measured, but you'd have to harvest the plant and refine the sugars out of them, distill them out in some way. It's going to take a long time. However, the oxygen produced is pretty easy, and there's some really simple ways to do it, and there's some harder ways to do it that we used to use, but now we have probeware that makes it a lot easier. So, measuring oxygen. Oh yeah, it's not glucose. So before we go any further, let's think about some experimental design concerns. What is the independent variable going to be in this experiment? Think about the setup that I've just laid out, the things that we could possibly manipulate. What's the independent variable going to be? Yep, it's going to be the sun, so the amount of sunlight. And what's the dependent variable? Well, we said we've got to look at one of the outputs of photosynthesis, and we said that glucose is hard to measure, so we are going to measure oxygen. And I included this particular photo because what we've got here, just to orient you, is we've got a beaker. This is not the way you're going to measure oxygen level, by the way, but this is the way that we used to do it before oxygen sensors back in the dark ages. We've got a plant that looks sort of like Elodia here, which if you had me for bio, you definitely used, and if you had anybody else for bio, you probably used. So we've got some Elodia here, and that Elodia is in water, and it's off its stem, it's creating little gas bubbles. And the way that we used to figure out oxygen production is we literally would count the number of bubbles for hours. Doesn't that sound like fun? That's not how we're going to do it. We're going to do it in a much cooler way. Enter the BIOCHAMBER 2000. It looks like this. Okay, it's not that cool. But instead of counting bubbles, we have an oxygen gas sensor. So you add an oxygen sensor and you've got a party. It's going to be super fun. I can't wait. So the general setup of the party is going to look like this, sort of, except that we're going to be outside. So here they've got a light ring set up around as their light source. We're going to use the sun. And for our dark experiments, we'll be covering up this chamber, probably using the cardboard dividers that you use when you take tests. So with that, I think you have enough information to go and, one, figure out what a null hypothesis is and why you might want to have a null hypothesis. And two, actually, two and three. Two, I want you to figure out a null and an alternative hypothesis for this experiment and bring them to class when you come and we see each other in person. And three, you should have enough information to come up with a numbered step hypothesis. So step one, do this. Step two, do this. Step three, do this. You should be able to have enough information to write a hypothesis that'll get you at least thinking about the lab in such a way that when you walk into class, if it's a nice day the next time we see each other, you will be able to just go out to the field with me and we'll be able to have our experiment. Make sure that when you come to class, your laptop is charged. If it's not charged and we're out in the field and it dies, we are not going to be able to collect data. So make sure that your laptop is fully charged when you come to class, and I'll see you then. Take care.